Okay, so a biomolecule, also called a macromolecule, is a molecule that is synthesized by living things. Right? It's primarily consisting of a carbon, and now carbon has the ability to form four electric bonds here, and that's going to give some stability and rigidity to most of these molecules. Right? So the primary feature, you can also have oxygen, you can also have hydrogen and nitrogen. And again, you can have sulfur and some other things, but these are the primary ones. Carbon being the primary component, and oxygen, hydrogen, nitrogen, giving us four basic types of biomolecules: carbs, lipids, proteins, and nucleic acids. Now we're going to go through these individually, and again, this should be a review for you. So we're going to go through at a pretty rapid pace. Okay? So let's start by looking at carbohydrates. These are composed of carbon, hydrogen, and oxygen. And these are basically links of carbon monomers. So a single carbon monomer is called a monosaccharide. Examples of this are glucose, galactose, and fructose. And these are basically, a monomer is a single part of several links that form a chain. So if you have one single monomer here, it's a monosaccharide. Disaccharides are where we start to group these together or add together. And this is where two monomers form a covalent bond together. So examples are lactose, where galactose and glucose are combined. Sucrose is another example, your table sugar, where glucose and fructose are combined. Lastly, we have polysaccharides. Do you have a question, Cheryl?
Could you repeat that one more time? You said, okay, monosaccharide, disaccharide, they form a double bond?
A covalent bond. Mm-hmm. So two monosaccharides form a covalent bond. What is a covalent bond? What is a covalent bond?
It's a bond where they share electrons.
Perfect, right? So it's a molecular bond where two elements come together and have shared electrons. Lastly, we had polysaccharides here, which was several monomers, right? Several different links of your monosaccharides grouping up together to give you this more complex group of carbohydrates. Examples of this are your starch, your cellulose, glycogen is an example. Where do we see glycogen? Where do we see glycogen in the body? Glycogen is, right? Not so much. But what is its purpose in the body? Where do we see it? Primarily muscles. Muscles, right? So it's going to be a storage of energy. You have lots of glucose and you want to put some away for later on. You can store that in the form of glycogen. Now let's look at the way that carbohydrates are synthesized and are broken down. We're going to look at two unique processes. So in order to be linked covalently, there's a process called condensation. And this is the joining of monomers of monosaccharides together, and this is going to release a byproduct of water, like H2O is going to release. Where do you see condensation happening like in everyday life? Some examples of condensation. When you breathe, yeah, you can see some condensation. What types of surfaces? On your mirror, on your glass, you can see some condensation. And that's basically where water is being produced. And in this instance, we're joining two monosaccharides or two molecules together and we're releasing water. The example here is that glucose and fructose come together, they release water, and then they form sucrose. Alternatively, there's hydrolysis. Now, hydrolysis is the opposite. This is where we split two larger saccharides apart into two smaller components, and we utilize water for this process. Okay? So in order for sucrose to be split again into glucose and fructose, we're going to need water as a part of that reaction, but it needs some water here. All right, so we add water, and then you can split it off into glucose and fructose again. This is called a hydrolysis reaction. Hydro meaning water, Lysis meaning to break or to cut or to cleave. Okay, hydrolysis. All right, so that's your carbohydrates, 3 groups, pretty simple. Your monosaccharides, disaccharides, polysaccharides. Let's move on to lipids. Lipids are an important group of molecules. Very important in terms of energy storage, important in our diet, and things like that. We have some essential fatty acids that we cannot make on our own. And they're basically composed of hydrogen, carbon, and sometimes a little bit of oxygen, not very much oxygen. Right? Not very much oxygen. What do we know about oxygen? What do we already know about oxygen? What is a feature of oxygen? How does it behave in terms of its anyone? So oxygen is an electron hog, right? It doesn't like to share electrons. Okay, so molecules that have a lot of oxygen are very partially, what we call partially charged. They have this polarity to them, where there's a partial charge around the molecule. Here we have one end that has a partial negative charge, one end has a partial positive charge, and this creates a polarity around those types of molecules. We see that with proteins, we see that with carbs, we see that with nucleic acids. Lipids are unique in that they are not polar. Okay, so they do not have a lot of oxygen, and as a result, they do not have this partial charge around the molecule. They have a nice, even, steady charge. They're what we call charge neutral. Okay, and this makes them non-polar. Now, what does non-polar mean in terms of its function? What is something that's non-polar? How does it behave? Anyone? What does non-polar mean with regards to water? Come on. It doesn't like water. Thank you. So it does not like water. It does not play nicely with water. This is where we get that oil and water phenomenon, right? You use the analogy that someone is like oil and water. They don't get along. They don't play well together. Oil does not mix with water for this very molecular reason. It is lipids are non-polar, okay? Because they do not have a lot of oxygen. And as a result, we also say that they're hydrophobic. Hydro meaning water, phobic meaning a fear of. Right? So non-polar being hydrophobic. Now there are 5 classes of lipids: triglycerides, ketones, phospholipids, eicosanoids, and steroids. And we'll go through them individually. So triglycerides usually are formed by a glycerol backbone. So we can see here in this orange, so this glycerol backbone, and then it has 3 fatty acid tails attached. And this is the structure of a triglyceride. Right? Now, the glycerol, the fatty acid chains that attach can be either saturated fatty acids or unsaturated fatty acids. What do I mean by saturated versus unsaturated?
Double bonds.
Double bonds, right? Double bonds where? Between the carbons, right? So if it's saturated, there are no double bonds between these carbons. Point that out here. So in this top one, this is saturated. None of these carbons are attached by double bonds, okay? And that means that this molecule, or these fatty acids, have as many hydrogens as is possible bound to it. So we see that they are saturated. Alternatively, unsaturated fatty acids are linked by double bonds. Now you can have monounsaturated, which has 1 double bond in the case of this fatty acid here. Or you can have polyunsaturated fatty acids, which have several double bonds. Okay, we're not going to get into the detail of that, but we do want to know that saturated means there's a presence of double bonds. There's not the presence of double bonds, excuse me, and unsaturated means that there is the presence of double bonds. Now, unsaturated fatty acids can also be cis or trans. What do I mean by cis or trans?
Cis is like kind of like mirrored. The trans would be on the opposite ends.
Okay, perfect. So cis or a cis configuration is where those hydrogens are on the same side. Okay, as we can see here, the example is oleic acid. This is a cis fatty acid. At the bottom is aldehydic acid, which is an example of a trans fatty acid. Okay, and that's where the hydrogens are on alternative sides of that double bond. Now, it's easy to think that, okay, both of these fatty acids have the same elements attached to it, and they're pretty similar, right? You just have that difference in location of the hydrogen. So both of these should, in theory, act the same way, have the same function, right? It's easy to think that, but that is not the case, right? That is not true. Why is that not true?
The body has certain enzymes that can only do things a certain way. Like with trans fats, the body can't break them down the way that they can regular fats.
Okay, perfect. That is true. Well said. All right. So what's unique is that this theory that I've been talking about, where form follows function, is true on a whole body perspective, but it's also true at a molecular level, right? So the way something looks, even the smallest, minute molecular detail is going to greatly alter the function of this molecule. Okay? So much so that trans fatty acids are said to be unhealthy, whereas cis fatty acids are not so unhealthy. Right? Trans fatty acids and saturated fatty acids are usually the source of what we call plaque formation, which can lead to stroke and heart attack and that sort of thing. So that smallest minute detail changes this molecule so much that it makes it from a healthy fatty acid to an unhealthy fatty acid. Okay? Everyone understand that? All right. Moving here. Next we have ketones. Right? Ketones. And ketones is where we have a carbon doubly bound to an oxygen. And you can have some sort of functional group attached here. So these R's at the bottom are representing examples of different functional groups. We have different types of ketones. The example here is acetoacetic acid. Okay, now ketones are also the byproducts of fat metabolism. So let's say you don't have enough glucose in your blood and you switched over to burning fats. The triglycerides in your fat tissue is broken down. Those free fatty acids can then enter the blood. They're going to go to the liver and they're going to be converted into energy, and ketones are going to be byproduct. Right? So ketones will build up in the form of ketosis, and this can lead to a state that's called ketoacidosis, because ketones are acidic, and so if they build up in excess in the blood, they can put the body into this acidic state, which is unhealthy, which is disease. It's leading to death, can lead to death, can be life-threatening. All right, this is seen in diabetics a lot. Okay, sometimes you have lots of sugar in your blood, but you don't have insulin, so that sugar can get into the cells to be used. And so you build up the breakdown product of fats, which is ketones. Now this is also associated with what we call an acetone breath. Right? That's the unique odor that's given off by persons who have lots of ketones in their blood. Now, I have a question.
Does it smell like fruit?
It does smell kind of fruity, yeah. A little bit fruity. Sometimes they call it a fruity breath odor. I'll also mention here, there are some diets, or there's recently been a diet fad, that's the ketogenic diet or the keto diet, keto-friendly diet, where persons intentionally limit or avoid carbohydrates in an attempt to shift their body to breaking down primarily fats as a source of energy. Okay, this diet can also be helpful in certain disease process.
Yeah. Doesn't this diet make it so you aren't having as much glucose? Doesn't the brain run on glucose?
So you do break down fat as your source of energy. So you can sustain yourself on this fat, but you do need some carbohydrates. Yeah, I wouldn't say that it's the healthiest diet. I wouldn't, advocate for removing any, total amount of food group from your diet, but some disease processes do benefit from being on a ketogenic diet. Okay. Any other questions on that bit? All right, so let's keep moving here. So the next class of lipids are phospholipids. Phospholipids. And phospholipids are unique in that they are amphipathic. Does anyone know what amphipathic means?
Polar and non-polar.
Right, so it has both polar parts and non-polar components. If you look at the structure of a phospholipid, it has a polar head and non-polar tail. Okay, what does polar mean? Sort of just talked about it, but what does it mean in terms of water, once again?
The water, the water.
Right, so polar means that it likes water, plays well with water. So the head of these phospholipids are hydrophilic, meaning that they like water, and the tails are hydrophobic or non-polar, meaning that they do not like water. Now, there's lots of water in our body, so this is going to make for some very interesting interactions. Right? Now, phospholipids create unique structures. One is going to be the phospholipid bilayer. This is the outer lining of our cells. This is the plasma membrane. It's also the lining of some of our organelles, such as the mitochondrial membrane, and so on. And what it does is it creates a very unique environment where the center of that lining is hydrophobic, meaning that it does not want to interact with water. Those tails, remember we said they were hydrophobic. And then the outer components are exposed to the aqueous environment around, either in the cell or out of the cell. So this structure really helps to create a barrier between different fluid compartments, right? So our intracellular fluid would be inside the cell, and our extracellular fluid would be everything outside the cell. So it's going to help to compartmentalize different parts of the body. Another unique structure that we see with phospholipids is called the micelle. The micelle. And this is basically a droplet that has a similar configuration where the phospholipid tails are facing each other to the center of that sphere, and the heads are facing the aqueous environment around. When I say aqueous, I mean it's just exposed to the water. Okay, so this is important in terms of transporting lipids, transporting other molecules, and this is actually what gives milk its white appearance. When you remove the fat from milk, you actually remove some of these droplets, and so that's why skim milk is a bit more paler in the color than whole milk. Oops, so there is a tidbit here I'll mention about surfactants. Now, another type of phospholipid is found in the lungs. It's called surfactant, and it's important in keeping the patency or keeping the airway of the lungs and of the respiratory airway open. Right, so there's a bit of water that's lining the lungs and the respiratory airway, and naturally what it wants to do is sort of interact with each other. That's going to cause the airway to collapse as both of those water linings want to interact with each other. And so surfactant is going to prevent that. We'll talk more about surfactant when we look at the respiratory system in YouTube. Then we have eicosanoids. Eicosanoids, we'll gloss over this very quickly, not as important. These are modified fatty acids. This is where an enzyme that's present influences these fatty acids to sort of fold upon themselves. And this creates a 5-carbon ring and 2 side chains. Okay, so you can see that 5-carbon ring on the left and then the 2 side chains on the right. Examples of this are prostaglandins and thromboxanes. Thromboxanes are going to be important in terms of the clotting cascade. Right? So they're going to be important in terms of the steps that allow blood to clot. And prostaglandins are important signals in the immune system. Steroids. Our last group of lipids are steroids or steroids. And these are derived from cholesterol. So some examples are estrogen, testosterone, aldosterone, we have cortisol, and these are important in the reproductive system as well as important in the endocrine system. You have steroids being produced in the testes, in the ovaries, in the adrenal glands. Now, the structure of steroids is that they are a ring-like structure made mostly of carbon and hydrogen.